Okay, ladies and gentlemen, you are welcome to my YouTube channel. Please don't forget to subscribe and join the YouTube community. So in today's video, we are going to look at the concept of gametogenesis. So what is gametogenesis? Starting with the introduction. Gametogenesis is the biological process by which reproductive cells called gametes are formed. So it's a biological process in which reproductive cells, sperm and egg cells are produced, and these gamete cells are very important in reproduction. So that is why it is called reproductive cells. So now, where this gametogenesis takes place? Gametogenesis occurs in the gonads of both males and females. And the primary purpose is to produce a haploid cells for sexual reproduction. A haploid cells, that is sperm cells and egg cells, are produced through gametogenesis, and these two gamete cells unite together through a sexual intercourse for reproduction purpose. And in the process of this gametogenesis is divided into two. We have what we call spermatogenesis, that is the synthesis of sperm. And then we have oogenesis. So this spermatogenesis is divided into phases. There is a reproduction phase, development phase, maturation phase, and then lastly, formation. So these are the two phases. Sorry, these are the four phases of gametogenesis. So you shouldn't forget about these four stages or four phases: reproduction, development, maturation, and then formation. And these four stages or four phases include the first stage, mitosis. As we can see clearly from here, it involves mitosis, then first division of meiosis, and then second division of meiosis. So these are the two phases. So these are the stages, or these are the events that occur at these four phases: reproduction, development, maturation, and then formation. So first, from the primary cells or from primary germ cells develop into first order oocyst, and spermatocysts, and then the second order spermatocytes, followed by spermatitis, and then lastly the formation where we have the spermatozoa. And also apply through in oogenesis, where it started with the primary germ cell, the first order oocyst, the second order oocyst, and then of course the ovulation, where there is a formation of a polar bodies or an egg cell. So these are actually the phases of the spermatogenesis, and we are going to look at the explanation as we continue. So the general overview of gamete formation. So gametogenesis, it involves meiosis, a special type of cell division. So this formation of gamete, it involves a meiosis, which is one of the special type of cell division. Remember, cellular division is divided into two. So we have meiosis and mitosis. So gametogenesis, it majorly involves the use of meiosis. So this gametogenesis, it's usually results in a four genetically diverse haploid cells from a single diploid precursor. So in gametogenesis, at the end, it involves the formation of four genetically diverse haploid cells. And these four genetically diverse haploid cells came from a single diploid precursor. And of course, this process of gametogenesis, it ensures the genetic variation in offspring, because this gametogenesis at the end, it involves the crossing over. And this crossing over, it ensures the genetic variability among offspring of the same parent. So this is actually the spermatogenesis and the oogenesis. And as we can see at the end, like for example, spermatogenesis, it ends up producing 4 genetically diverse haploid cells. You see it come from a single diploid cell. This is a diploid cell. This is a diploid cells. And at the end, we end up having 4 genetically diverse haploid cells. So the process is called spermatogenesis. These are actually the difference between spermatogenesis and oogenesis. So at the end, as we can see, the first step, that is N1, where we have two, and that is a diploid cell segregate into a haploid cell. So the first step is called a phase polar body. And then the second phase is called the second, where we have N also in the case of spermatogenesis, is spermatids. And then third, which is the end product, is called sperm. And then the second polar tooth, which is the N also from the zygote, and then from the sperm, then they will come up together. After the fertilization, then a zygote will be generated. So what I'm trying to say, ladies and gentlemen, in the process of gametogenesis, it involves the formation of 4 genetically diverse haploid cells from a single diploid precursor, which of course at the end fertilize to produce a zygote. So then let's look at the spermatogenesis in males. So spermatogenesis occurs in the testes of males, and it begins at puberty and continues throughout adult life. That is why it started. So it started from puberty, that is uh, at the level of maturity, and it continues throughout adult life. So it means that once you are matured, so there's a likelihood that spermatogenesis will continue throughout life, especially in the case of males. So the process produces sperm cells. So at the end of spermatogenesis, sperm cells are generated or are produced, or spermatozoa, for fertilization. So as we can see, this is where we have a spermatogenesis, where it started from, look at it, it started from A, where we have number 1, it started from spermatids here. And then from spermatid, we have a germinal cell, where the sperm cells are generated. Then we have epididymis. What is epididymis? Look at it, as we can see, 3. This is epididymis, where the sperm are stored. We have vas deferens, testes, coiled seminiferous tubules, and many more. But at this point, we are talking about spermatogenesis, which usually takes place in the testes of males. And at the end, it produces sperm cells or spermatozoa, which use for fertilization. So then the next thing is oogenesis in females. So oogenesis takes place in the ovaries of females, and it starts before birth. So see, at this point, the formation of oocysts or egg is started from birth. So it started before giving birth, but is completed only upon fertilization. So see, that is the difference between the spermatogenesis and oogenesis. Spermatogenesis started at the stage of puberty and continues throughout life. But for the oogenesis, it actually starts before even giving birth, and it's completed upon fertilization. So this process, it results in the formation of a single ovum or a polar bodies. So you see, in the case of spermatogenesis, at the end, you produce a four genetically diverse cell. But in the case of oogenesis, only a single ovum is formed. Look at it. It started from oogonium, where we have 2n, then after mitosis, we have 2n. That is where we have E. So from oogonium, after mitosis, we have 2n, and that is where we have a primary oocyst. And it arranged in phase 1. Then we have meiosis will now continue after puberty. And then at the end, a secondary oocyst where it arrested in metaphase 2. Then we have ovulation and sperm entry, where we have a meiosis again with fertilization, and then a fertilized egg or zygote or polar tooth will be generated. So this is actually oogenesis. So what are the stages of spermatogenesis? So spermatogenesis, it includes spermatogonial mitosis, meiosis, and then spermiogenesis. So this, of course, this spermatogonia is divided mitotically to maintain the germ cell line. And of course, the meiosis reduces the chromosome number and produces 4 sperm cells from each spermatogonium. So you see, in the case of spermatogenesis, 4 sperm cells are produced from a single spermatogonium. But in the case of oogenesis, only one ovum is generated. So as we can see, this is the different stages of spermatogenesis, where the spermatogenesis, as we can see, it started between 15 to 17 days, that is spermatocytogenesis. The meiosis also for another 15 to 17 days. Then at the end, we'll have a spermatozoa. So as we can see, from a single spermatogonium, at the end, the 4 sperm cells are generated, and it involves, as we can see, mitosis, meiosis 1, and meiosis 2. Before at the end, a spermatozoa or sperm cells are generated. So what are the stages of oogenesis? So oogenesis, it involves the development of primary oocysts within ovarian follicles. So first thing, the primary oocysts are produced or are developed within ovarian follicles, and then it includes meiosis 1 and meiosis 2, which are arrested at specific stage until fertilization. So this is where we have the oogenesis is started from primary oocysts. And the primary oocyst will develop and then maturate. Then we have a secondary oocyst or polar tube. And finally, a mature ovum will be produced. So the process results in a large ovum and polar bodies that degenerates. So at the end, a large ovum will be produced and a polar bodies. So these are the end product of oogenesis, a large ovum and 3 polar bodies. So what are the hormonal regulation of gametogenesis? Gonadotropin hormones like FSH, that is follicle stimulating hormones, and LH, which is luteinizing hormones, regulate gametogenesis. So these are the major hormones that are involved in regulation of gametogenesis. In males, these hormones, FSH and LH, stimulate spermatogenesis in the testes. So they stimulate gametogenesis in the testes, and then in females, they control the maturation of oocyst and ovulation. So you need to understand this. In the male, it stimulates spermatogenesis in the testes, while in females is for maturation of oocyst and ovulation. So these are the two hormones, LH and SH. They are produced by the hypothalamus and they carried out their function at this point in the testosterone. In the testes. So then, genetic variation throughout gametogenesis. So usually during gametogenesis, there is a crossing over. And this crossing over during meiosis promotes genetic diversity. And independent assortment of chromosome contributes to variation in gametes. So you see, during this oogenesis, there is meiosis, and this, of course, at the stage of meiosis, there is an issue of crossing over, especially when the homologous chromosome are aligned. So this crossing over that takes place between one chromosomes to another, it promotes genetic diversity. And also in addition, this genetic diversity through gametogenesis, because there is an issue of independent assortment of chromosomes, because chromosomes are assorted during gametogenesis or during fertilization. So this assortment of chromosome, it also contributes to variation in gametes. So this diversity increases the adaptability of species over generations, because environment are changing and all living organisms are trying to adapt and survive in the environment. So as a result of that, living organisms always tend to undergo the genetic variation. So what are the things that contribute and promote genetic diversity or genetic variation? Is the crossing over and then the assortment of chromosome. So as we can see, crossing over during meiosis increases genetic diversity. Crossing over is the exchange of chromosome segments between homologous chromosomes. Look at it, ladies and gentlemen. We have at the first stage here, we have two homologous chromosomes. So these two homologous chromosomes pair off with each other during prophase 1 in meiosis. So during prophase 1, they will align or pair off with one another. So this is what happened here. So in this position, some chromatids are very close to each other and segment cross. And at the end, this is what we are going to have. The homologous chromosomes exchange the segment of the DNA between the homologous chromosomes. So some of these segments will break off and reattach to the other homologous chromosome. So this is the crossing over. So it occurs during prophase 1. So what are the clinical relevance of gametogenesis? Abnormalities in gametogenesis can lead to infertility or genetic disorders. Techniques like in vitro fertilization rely on understanding this process. So if you're able to understand this process, then you can easily do in vitro fertilization. What is in vitro fertilization? It usually happens when there are two couples that are infertile. One is normal and the other is infertile. So you can get the sperm and the egg cells and then you can fertilize them in test tube or in glass. That is what you call in vitro fertilization. You carry out or you do fertilize sperm and egg cell in test tube or glass. So research in gametogenesis also aids in reproductive technologies and genetic studies. So then, what are the summary and conclusion? So gametogenesis is essential for sexual reproduction and genetic diversity, and it involves complex processes of meiosis and cell differentiation. So understanding gametogenesis is crucial for advancement in medicine, reproductive health, and genetics. Ladies and gentlemen, thank you very much. Don't forget to subscribe and join my YouTube communities for more videos.